Hello students, welcome to EPG Patashala. I am Dr. Gayatri, working as Assistant Professor in Center of Advanced Study in Crystallography and Biophysics, University of Madras. Today we are going to talk on module Hydrogen Bonds in the paper Biocrystallography. So students, let's see what are the objectives of this model. First we'll see what is a hydrogen bond, various perspectives of hydrogen bond, properties and classifications of hydrogen bond, and the importance of hydrogen bond in structural chemistry and structural biology. In the year 1939, Pauling gave a definition of hydrogen bond that under certain conditions, an atom of hydrogen is attracted by strong forces of two atoms instead of only one, so that it may be considered to be acting as a bond between them. Hydrogen bond is a special case of dipole-dipole attraction, which is much stronger than the van der Waals interaction but weaker than the covalent and ionic bonds. The larger the dipole moment of the hydrogen bond, the stronger will be the hydrogen bond interaction with the acceptor. Also, strong hydrogen bonds occur if the donor and the acceptor atoms are highly electronegative. This picture shows the anatomy of the hydrogen bond. The bond between the donor and the hydrogen atom is polar covalent bond. The hydrogen atom forms an electrostatic attraction with the neighboring electronegative atom, which acts as an acceptor, thereby it forms an hydrogen bond. T.S. Moore and T.F. Windmill were the first to mention about the hydrogen bond in the year 1912. In order to give the evidence of the hydrogen bond, we compare the boiling points of methane, ammonia, water, and hydrogen fluoride. We see a greater variation for these similar sized molecules than what is expected. Most of the simple hydrides of group 4, 5, 6, and 7 elements display a tremendous rise in the boiling point with molecular mass, but the hydrides of most electronegative elements like nitrogen, oxygen, and fluorine have abnormally high boiling points for their mass. This exceptionally strong dipole-dipole attraction that causes this strange behavior is called as an hydrogen bond. Hydrogen forms a covalent bonds to more electronegative atoms such as oxygen, and because a hydrogen atom is quite small, the positive end of the dipole, that is a bond dipole, can approach neighboring electrophile or basic sites more closely than other polar bonds. Coulombic forces are inversely proportional to the sixth power of the distance between the dipoles. Based on this principle, the hydrogen bonds are relatively strong when compared to the van der Waals interactions. The unique property of water is largely due to the strong hydrogen bond that occurs between these molecules. Hydrogen bond is an interaction between the hydrogen atom in a polar nitrogen-hydrogen, oxygen-hydrogen or fluorine-hydrogen bond and an electronegative oxygen-nitrogen or fluorine atom. The high electronegative difference of NH, OH and HF bonds cause these forces to be very strong than the van der Waals forces. Before we proceed further, we should learn few terms like the electronegativity and what is dipole-dipole interaction. Electronegativity is a tendency of an atom in a molecule to attract the bonding pair of electrons to itself, thereby it forms a dipole and form the bonds. In other words, electronegativity is a tendency that tells how strongly the atoms attracts the electrons in a bond. If the electronegativity of an atom is high, then it attracts electrons. The difference in electronegativity determines the bond type and also it explains what is meant by a polar bond and a polar molecule. The high electronegative difference of NH, OH and HF bonds causes these to be stronger forces than the van der Waals forces. Before we proceed further, we will see what is a polar atom. Polar atom is an atom which has one end that is positively charged and the other end negatively charged. In the case of a water molecule, where oxygen is highly electronegative, the electron cloud of hydrogen atom will be distorted towards the highly electronegative donor atom, so that the thinly shielded positively charged hydrogen nucleus becomes strongly attracted by the neighboring electronegative acceptor atom. Thereby, the oxygen will have a partial negative charge and hydrogen will gain a partial positive charge. These partial charges on oxygen and hydrogen atoms will lead to a hydrogen bonding network in water solvent by dipole-dipole interaction. In dipole-dipole interactions, the hydrogen bond is a weak attraction between a partially positive hydrogen in one molecule and partially negative oxygen in the another molecule, thereby creating a dipole-dipole interaction. So the dipole-dipole interaction is an attractive force between positive end of one polar molecule and with the negative end of the another polar molecule. Classical hydrogen bonds will occur when the hydrogen atoms are bound to a highly electronegative atoms like nitrogen, oxygen or fluorine. Nitrogen has one lone pair of electrons, oxygen has two lone pairs of electrons and fluorine has three lone pairs of electrons. Therefore, there are two important conditions for the formation of classical hydrogen bonds. This picture shows the energy of the hydrogen bond. Based on the energy of the hydrogen bond, they are classified as strong, moderate and weak hydrogen bonds. In general, the energy of hydrogen bonds vary approximately from 8 kJ per mole to 40 kilojoules per mole. It is very clear from this energy chart of various bonds and interactions that the hydrogen bond is weaker than the covalent and ionic bonds, but it is more stronger than the van der Waals interactions. If we look into the bond energy of fluorine, hydrogen and fluorine, the bond energy is 161.5 kilojoules per mole. Whereas in the case of nitrogen, hydrogen and oxygen as a donor hydrogen and acceptor atom, the bond energy is approximately 8 kilojoules per mole. In order to categorize the hydrogen bonds as strong, moderate and weak, the distance between the donor and the acceptor atom is very much important. And the distance between the donor and the acceptor is considered as a bond length between donor and acceptor. Experimentally, bond length is in general shorter than the sum of the donor hydrogen bond length and the van der Waals radii of hydrogen and the acceptor atoms. The HA distance is not in all hydrogen bonds shorter than the sum of van der Waals radii. For a donor hydrogen group to be able to form a hydrogen bond, the donor doesn't need to be very electronegative always, but it is only necessary that the donor hydrogen bond is at least slightly polar. Also, in the case of hydrogen acceptor, the counterpart acceptor doesn't need to be particularly electronegative atom, but it has to supply the sterically accessible concentrations of the negative charges. Hydrogen bonds are generally classified as strong, medium and weak depending on donor acceptor distance, hydrogen acceptor distance and the angle subtended at the hydrogen atom. Hydrogen bonded complexes are generally stabilized by electrostatic, induction and the dispersion energy terms. The electrostatic term contributed by the dipole-dipole and ion-dipole interactions gives the hydrogen bonds their highly directional nature. In the case of weak hydrogen bond, the hydrogen bond is an interaction wherein a hydrogen atom forms a bond between two structural moieties, that is a donor and the acceptor. In this case, the donor is given with a symbol X, of which one or even both are of moderate to lower electronegativity. In order to get a complete idea and more details about the hydrogen bond, one can refer the book by Professors Desiraju and Steiner, the book titled as The Weak Hydrogen Bond in Structural Chemistry and Biology, published in the year 1999. This graph shows the distinction between strong and weak hydrogen bonds. In the case of OHO, hydrogen bond distance, that is the distance between the donor and the acceptor, varies from 2.3 to 2.8 angstrom, which is a strong hydrogen bond. Whereas in the case of CHO hydrogen bond, the distance between the donor and the acceptor atom varies from 3 to 3.8 angstrom, which is much weaker when compared to OHO hydrogen bond. So the strength of the hydrogen bond depends on various parameters like the direction, length, and the angle between donor hydrogen and acceptor. Hydrogen bonds are highly directional and they are generally asymmetric. The bond strength depends on the angle and the length. The strongest hydrogen bonds are generally linear and the strength decreases exponentially with the distance. Also, the strength of the hydrogen bond depends on temperature, pressure, and the environment. Let's see the various perspectives of hydrogen bond by different scientists all over the world. Pauling, in the year 1939, he gave a definition of hydrogen bond as we saw very early at the beginning of this module that under certain conditions, an atom of hydrogen is attracted by rather strong forces to two atoms instead of only one, so that it may be considered to be acting as a bond between them. Pimentel and Macklin definition in the year 1916 says that a hydrogen bond is said to exist only when there is an evidence of the bond and there is an evidence that this bond specifically involved a hydrogen atom which is already bonded to another atom. Desiraju and Steiner in the year 1999 said that weak hydrogen bond is an interaction wherein a hydrogen atom forms a bond between two structural moieties, that is donor and the acceptor, of which either one or even both are of moderate to low electronegativity. Moving on to the geometry of hydrogen bonds, there are different types of hydrogen bonds. There are two-centered linear hydrogen bonds which are more stable than the three or four-centered hydrogen bonds. Three-centered hydrogen bonds are called as a bifurcated hydrogen bonds. Let's consider the example of water molecule where two hydrogen bonds acts as a donor and two are accepted, which is in the case of a bifurcated donors and acceptors respectively. Four-centered hydrogen bonds are also possible where donor or the acceptor will be trifurcated. In the case of CH-pi interaction, the CH bond generally has a dipole moment and this hydrogen bond comes from the interaction between positively charged hydrogen atoms and the negatively charged pi phase of aromatic system. There are different types of CH-pi interactions. To name a few here are T-shaped, which is nothing but edge to face hydrogen bond, edge tilted T and face tilted T. Next we move on to intra and intermolecular hydrogen bonds. Intramolecular hydrogen bonds are present within the molecule and they stabilize the molecular structure, whereas intermolecular hydrogen bonds are present between different molecules which stabilize the crystal packing as well as the crystal structure. Let's have a look on patterns, rules and selectivity of hydrogen bonds. Margaret Ada Eton Kennan focused on the patterns of hydrogen bonds in the aggregates of small organic molecules using different graph sets. She recognized the need for a general and simple method for characterizing, recognizing and comparing the patterns of hydrogen bonds in molecular aggregates. By using the graph theory to molecular aggregates, she broke down the complex networks involving different types of hydrogen bonds into simpler constituent motifs. That is, all patterns of hydrogen bonds can be broken down into just four simple motifs, that is chain, ring, dimer, self. The pattern formed by molecules connected to one another by one type of hydrogen bond is called as a motif, which is characterized by these four designators as we saw: chain, ring, dimer and self. Self means intramolecular hydrogen bond. Here R228 hydrogen bond is shown. This R represents a ring motif. The 2 in the subscript denotes number of donors involved in forming the ring. The 2 in the superscript indicate number of acceptors involved in the formation of the ring motif. And the number which is given in the bracket, that is 8, it indicates how many atoms are involved in the ring pattern. Intermolecular hydrogen bonds in crystal packing generates different beautiful patterns. It's not only in structural chemistry that the hydrogen bonds stabilize a molecule and crystal packing, but also it has wide significance in structural biology in terms of macromolecules. Moving on to the significance of hydrogen bonds, we all know that hydrogen bonds holds 2 strands of double helix together. It holds a native polypeptide chain into a stable secondary structures. It helps the substrates to bind with their enzymes. It helps antibodies to bind to their antigen. Hydrogen bonds also contribute for the structural stability and also the folding of the molecules. It contributes for the catalysis and also in protein ligand, protein-protein and protein-DNA interactions. In the case of hydrogen bonds in DNA, hydrogen bond plays an important role in determining the three-dimensional structure that is adopted by macromolecules, where the bonding between certain parts of a molecule causes it to fold into a specific shape, which in turn helps in determining the molecule's physiological and as well as the biochemical properties. Let's consider the example of double helical DNA structure, which is formed by the hydrogen bonding between its base pairs and also by pi stacking interactions that link one complementary strand with the other. In general, we can say that DNA is a polymer of nucleotide linked together by phosphodiester bridges. As we all know that hydrogen bonds hold 2 DNA strands together. Phosphate group of one nucleotide bonds with the hydroxyl group of the following nucleotide, thereby generating a single strand of DNA molecule. DNA double helix is made up of 2 polynucleotide strands intertwined together to form a long helical structure. These 2 strands are held together and stabilized by hydrogen bonds between them. The nucleotide adenine pairs with thymine and guanine pairs with cytosine. The pairing occurs through the hydrogen bond. Between adenine and thymine, 2 hydrogen bonds are formed, and between guanine and cytosine, 3 hydrogen bonds are formed. In case of protein structures, the function of any protein is determined by its three-dimensional structure, which in turn is determined by the different types of bonds that stabilize the structure. Hydrogen bond is one of such bonds that stabilizes the secondary as well as the tertiary structure of proteins, and also it stabilizes protein ligand interactions and protein substrate interactions. Secondary structure of proteins like the alpha helix, beta sheets, the turns are created by the hydrogen bonds between the carbonyl oxygen and the amide hydrogen. In the case of alpha helix, different types of helices are described based on the number of amino acid residues per turn, the pitch and the distance per turn, which in general are decided by the nature of amino acids and also by the hydrogen bonding existing between them. When the spacing of amino acid residues participating in the formation of hydrogen bond occurs regularly between I and I plus 4 residue, then the helix is characterized as an alpha helix. If the hydrogen bond is formed between I and I plus 3, then a 310 helix is formed. When 2 strands are joined by hydrogen bonds involving alternating residues on each participating strand, a beta sheet is formed. In parallel beta sheets, hydrogen bonds formed are not linear and it includes 12 atoms in a ring pattern. But in the case of anti-parallel beta sheets, 10 to 14 atoms are involved in a ring motif generation, and the hydrogen bonds are linear and are more strong when compared with the hydrogen bonds formed between the parallel beta sheets. This picture shows the distinction between the anti-parallel beta sheets and parallel beta sheets. In beta turn, which includes 4 residues, the hydrogen bond is formed between the first and the fourth residue, where the central 2 residues don't participate in inter-residue hydrogen bonds. In the case of gamma turns, which include 3 residues, the hydrogen bond is formed between first and third residues. Hydrogen bonds play a major role in protein ligand interactions at the active site of the enzyme. The donor or the acceptor atom can be either from the amino acid residue at the active site, or it can be from the ligand atom which binds at the active site of the enzyme. According to Lipinski's rule of 5, in structure-based drug design, there should not be more than 5 hydrogen bond donors and not more than 10 hydrogen bond acceptors in a ligand molecule. This figure shows, the central image shows how the ligand binds at the active site pocket of the enzyme. And the Lick plot representation can be made to see the number of hydrogen bonds the ligand makes at the active site and also the hydrophobic interactions it makes with the amino acid residues at the active site of the enzyme. So students, let's now summarize what we have learnt in this module on hydrogen bonds. We saw that hydrogen bonds play an incredible role in providing the stability to the three-dimensional structure of small and macromolecules. The strength of the hydrogen bond is characterized based on the electronegative potential of the donor and the acceptor atoms. The strength of the hydrogen bond also depends on the distances between the donor hydrogen, hydrogen acceptor, and the donor acceptor atoms. It also depends upon the angle between donor hydrogen and the acceptor. In turn, it depends upon the environment. We also saw the importance of hydrogen bonds in the stability of macromolecules and also the functional importance of hydrogen bonds in protein ligand complex formations. Thank you.